welcome to our first interview of the OBBPS, the Oxford Brookes Biomed podcast series. Today, we will be talking with Dr. Dave Carter, a reader and lecturer at Oxford Brookes University. Dave graduated from York University with a bachelor's degree in biochemistry, which included a year working on the Human Genome Project at the Sanger Institute in Cambridge, a worldwide renowned institute and one of the largest concentrations of genomics and biodata in the world. He completed his PhD at Cambridge University under the supervision of Dr. Peter Fraser at the Babraham Institute. During his PhD, he developed a novel essay titled RNA Tagging and Recovery of Associated Proteins to demonstrate a physical interaction between a locus control region and the beta globulin gene in red blood cells. He then worked at Oxford University as a postdoctoral researcher in Professor Peter Cook's lab, investigating the structure of transcription factories. He was appointed as senior lecturer in biomedical science in October 2009 and has established a lab to study the effects of non-coding RNAs and extracellular vesicles in stress response to further understand and treat diseases such as cancer. I'm also joined by Chloe Sterling, the Vice President of our Society, and Priscilla Purat, our Academic Officer. And hello, Dave. Thank you so much for joining us. How are you?
Hello. Yeah. Okay. Thank you. Yeah. Can't complain.
Thank you so much for coming on board and for being our first guest on the podcast. And I guess we can dive straight into the questions. Chloe, you can take it away.
Okay. All right. First question. What was it like studying at the Sanger and Babraham Institutes, respectively?
Yeah, thanks. Thanks for having me. It's a pleasure to be here as well. Okay, so I'll try and keep it relatively short. So the Sanger, that was a great place to be, actually. So the Sanger, when I went there, it was back in 1998 was when I first went there, which I'm guessing is probably before you were born, which makes me feel old. 98, and that was at the height of the Human Genome Project. So it was really, really exciting at that time. I was just an undergrad, and I was like super shy, and I didn't know what was going on in the world. And then I got to do this year out working on the Human Genome Project, doing radiation hybrid mapping, which I won't go into the details of it, but it was really exciting. And so it was what was really special about that place was that you just felt this kind of everyone's working together towards this bigger goal, something much bigger than yourself. So it was really exciting, actually. And then I came back for another stint there in 2000, just before my PhD. So yeah, it was really nice. And when the T-shirt came out, the Human Genome Project T-shirt came out, I was on that T-shirt, so I was quite happy about that. And then I went to the Babraham Institute I started my PhD in 2000, in September of 2000, and that was a really nice place as well. And it's attached to Cambridge. It's affiliated with Cambridge University, but it's like a research intensive institute that's quite intensively funded by the BBSRC, which is one of the big funding councils in the UK. And yeah, so it was super exciting to be part of that, and the PhD itself obviously was a lot of fun. So it was a good time. Good times.
Fantastic. No, it sounds really interesting. I mean, to be part of such an enormous project, like you said, with the Human Genome Project, and I think like what you mentioned, I think that's a big target of a lot of young researchers now, to be trying to be part of a big picture. I think that's something really attractive, and I can imagine the adrenaline rush to try and think of, especially that's such a big advancement, especially nowadays, where we're basing a lot of future developments and work off your work, really, what you helped out in. So it's really good. And so just to go on to also your personal work a bit, can you give a brief outline? You mentioned this very briefly with your radiation work and explanation of your PhD project on how you investigated the beta globulin locus controls regions and its association with an actively transcribed beta globulin gene within the red blood cell, which is the main component in hemoglobin, and using procedures. The abbreviation is RNA FISH TRAP, which seems is a very interesting abbreviation, but it stands for RNA fluorescence in situ hybridization tagging. and the recovery of the associated proteins. And if you can just give us an idea of your personal work and the work at the Sanger Institute and Babraham.
Yeah, so that was my PhD work that you just described. During my year out, I did the stuff at the Sanger was radiation hybrid mapping. So these days we have the whole of the genome sequenced, but when it was being sequenced for the first time, before we had the full sequence, we needed a kind of set of signposts, genomic signposts that were saying this massive chunk of DNA belongs to this part of the genome. And so to do that, you first need a set of signposts to then look in that big chunk of DNA and see which of the signposts are in this big chunk of DNA, so that I can place this big chunk on the map. And so that's to do that. There are different ways of doing it, but the way that we were doing it was called radiation hybrid mapping. And as I said, I don't know the massive details of it, but the way that you do it is you're doing PCR polymerase chain reaction based markers to see in these panel of different of hybrids. So I'm going into it now. So you take these, I can't remember, they were hamster cells. It was a long time ago now. And you irradiate them so that the human cells, that's right, you irradiate the human cells, that the genome breaks up, and then you fuse them with hamster cells. And so different bits of chromosomes go together and get rescued in these different cell lines. And so you look at, I'm not explaining this very well at all, but never mind. And then you look for the different PCR-based markers to see which ones went together. And so the more often two markers go together, the closer they are together on the genome. And so by doing that with a bunch of lots of different markers to see which ones go together in these different panel of cell lines, you can figure out where they are relative to one another, how close they are relative to one another. And when you do that with thousands and thousands and thousands of markers, you eventually build up a map of where they all are. So that was quite fun. I mean, for my part, I was just at the bench doing a lot of pipetting, every day. And but as I said, it's exciting to be part of that project. For my PhD, yeah, that was a lot of fun actually. So for my PhD, as you described, we did that RNA FISH TRAP, which is a long time since I've heard it actually said out loud, the full acronym. But so the way that what we were interested in is essentially how genes are switched on or off, and how they're regulated. So all of your genes in your cells need to be regulated. They need to be switched on at the right time, in the right cell type, in response to the right stimulus, and switched off at the right time. And in the case of the beta globin gene, in red blood cells have to carry lots of hemoglobin, and the beta globin gene produces a protein which is 1/2 of hemoglobin, and in red blood cells you need tons of it. So you have to have a very, very active, highly switched on gene, beta globin gene, in that cell type. And one of the ways in which that is achieved is by this thing called the locus control region, which is a bit of DNA that's connected. It's all along the same stretch of DNA. So you've got this long stretch of DNA that's got the gene, and then about 30,000 bases away, you've got this other stretch of DNA, which is called the locus control region. And we knew already at the time, that it was needed to activate the gene. It's absolutely necessary to really activate the gene to a high level. It's a type of enhancer element. And so it enhances the expression of the gene. But what we didn't know was how. We didn't know how it was activating the gene. We didn't know whether it was physically interacting with the gene. So the enhancer is actually I can't quite see it on the screen, but if you imagine this long piece of DNA that's like the length of my arms, what we didn't know was whether they were looping across to touch each other, so to speak, and interact. And that's what activated the gene. Because at the time there were other labs that were proposing other theories, which were quite reasonable at the time. Maybe they're actually acting at a distance. And so you've got the enhancer over here and the gene over there, and there's some kind of signal that's sent along the length of the DNA to activate the gene. And so we developed a technique, which is the RNA FISH TRAP technique, which essentially was able to detect that interaction. And we showed that the two things come together and the region in between is looped out. And it's becoming increasingly obvious now that is the way that chromosomes are actually organized physically in space. Because the thing about packaging DNA into a cell, you've got 2 meters of DNA that has to fit into a teeny tiny cell that you can't actually see. So there's all lots and lots of folding and intricate folding that happens, and loops. and these kind of structures are really important. And in terms of gene regulation, that interaction is really, really crucial. So that's what we developed at the time as part of the PhD. So it was really exciting to be part of something like that, because it did answer a really important question at the time, and led to a nice paper and some other following work as well, which was good fun. So, yeah,
was the application of the RNA FISH trap, was it the first time it was being used with a gene such as beta globin? Was it used in the past with other locus controls, or was it really the first time in that context using this procedure?
It was the first time anyone had used this procedure at all. So,
oh wow, okay.
It was actually Peter Fraser, who's a great guy. Actually, he came up with the idea. So I was just a PhD student coming in and saying, "Right, boss, what project are we doing?" And he said, "Well, I've had this crazy idea. I don't know if it'll work, but let's try it out." So FISH, fluorescence in situ hybridization, is a technique that is used to essentially visualize when genes are present or switched on, depending on the type of fish that you do. It's RNA fish or DNA fish. It's a way of lighting up a gene or an RNA. And what he thought of to do, and I did the project, was to, instead of just lighting up for the sake of seeing it, let's light it up in a special way, which actually causes specific type of molecule to be deposited in the nearby region. So what we did was we tagged the active gene, and we deposited in the physical vicinity around the active gene this particular, I think it's called biotin tyramide. Doesn't matter. But the point is that it's this molecule that gets deposited on around the close vicinity to the active gene, and then you can use that as a handle to pull out the pieces of DNA, the pieces of chromatin that have this particular marker. And so what you can then do is, depending on how close the DNA was to the active bit that you labeled, you could see how close a piece of DNA was to the epicenter based on how much of this protein, based on how much of this marker it had on it. And so we're able to then essentially draw a map of what was nearby the active gene. And we could see that the bit in between the LCR and the gene was really absent. But then when you look to the LCR, suddenly you've got this big spike of signal, which tells you that, that this thing must have been really close to the active gene to get labeled in that way. So no one had done that before. No one had done that before.
Amazing stuff. Wow. That's really,
I think that's just for any scientist. For me, that's the beauty of the science. And whether it's an undergraduate project, whether it's whatever research it is, the beauty of it is that, and I think this is where the kind of real beauty of science is generally in doing research, is that when you get that result, you do the experiment, when you get the result, you're the first person in the world ever that has that result. Even if it's a technique that other people have used, you're going to be doing that experiment to ask a question which has not been answered before. Otherwise, you wouldn't be asking that question. And so when you get that result, even before you're a supervisor, you're the first person in the world who gets that result. And that buzz is actually, there are lows that go with doing science. There are lows, but that high is so, it's just amazing. So yeah, it was a good time.
Thank you so much.
So we understand that you ran the biology of cancer module in third year, which unfortunately we won't be getting. And honestly, personally, I'm like devastated because I loved your lectures last semester. But we understand that you've done some ongoing research about the effects of non-coding RNAs and extracellular vesicles in stress response. Could you please tell us a bit more about this, the basis of this research, and what you have found out?
Yeah, sure. So, yeah, I'm sorry, I'm going. I won't be doing that module anymore. I did enjoy teaching on that module. It was good fun. Alas, I'll be moving on. So, yeah, I've been interested in non-coding RNAs for quite a long time. So I guess just for those that might not know about it. So, non-coding RNAs are RNAs which are not turned into protein. So usually the DNA gets transcribed into RNA, the RNA gets translated into protein, and that is the way things work. And in the recent, last few decades, we've realized that actually there's a lot of RNA that is not turned into protein. It's the point of it is to be made into RNA, and then it does stuff. It's not coding for protein. And so I was fascinated by this for a long time. Actually, that was what got me into science in the first place. That's what really hooked me in, was this idea of non-coding RNAs. And so I was doing work on that. And there's different types. You get like long ones and short ones, long non-coding RNAs and then short non-coding RNAs. And I was particularly interested in long non-coding RNAs and how they're affecting DNA and chromatin structure, but also microRNAs and how they affect cancer progression, things like that. And so about, was it nearly 10 years ago now, time flies. So I was working with Professor Munira Kadim, actually from Brookes as well. And so she was working on this thing called the bystander effect. The bystander effect is nuts, it's crazy. So the idea is that if you have cells that get stressed, then you see evidence of that stress. You can see, if you hit them with radiation, for example, you can see DNA damage. You can see stress response in those cells, which makes sense because you've just stressed them. But what's crazy is that they also release something into the extracellular space. They release something out of the cell, which if it then goes into other cells, causes those cells to get stressed too. Which means that it opens a can of worms in a way that, you know, like why would you want to do that? So let's imagine that the four of us here are cells in a dish. If I get stressed, then what I'm doing is I'm going to then release something which is you're going to see it, you're going to pick it up, and then you're going to start freaking out and getting stressed too. So why would I want to do that to you guys? Because you seem like really nice cells. So why would I want to sort of make you feel my pain and my misery? And so but if we have time, I can get back to that point. But what I was interested in at that time, I'd never heard of the bystander effect, and I was interested in non-coding RNAs, and I saw her give her talk, and I saw one of her students give a talk, and then I just thought to myself, has anyone ever actually checked to see if an RNA molecule, because no one knew what that signal was, people knew that there was involvement of cytokines and various things, but it wasn't really clear what was causing this. And so I asked one of her PhD students after he gave a talk, has anyone checked if an RNA molecule is involved? And amazingly, no one ever had, really. So we took the way we do these experiments is you take your cells, you zap them, you let them sit there stewing in their own juices for 4 hours, 8 hours, whatever you want, and they sit there producing this signal into the liquid that you grow them in. You take that liquid off, and you then get rid of all the cells, so you've just got the signal that's left. And what we did was we treated that with RNAs to degrade any RNA molecules that were in there, and we lost the signal. We lost the ability of this conditioned media to induce stress in bystander cells. And then I got into extracellular vesicles. I was at a conference, I heard about them, someone gave a talk about them, and I straight away was like, oh, we've got to check if extracellular vesicles, EVs, are involved. And so we came back and we did the experiment, and they do seem to be involved. So it seems then that as a cell, if I get stressed, I am then producing these, as a cell, I'm always producing extracellular vesicles, right? EVs are super, super, super cool. I mean, I could go on about them. They're super cool. And what happens is when I'm stressed, I change what I'm putting into them. I'm still producing them, but I change the composition of them. And so EVs used to be people used to think of them as hey, a cell is just using them to get rid of rubbish. But now we know that they're a part of the signaling that goes on between cells. And so as a cell, if I get stressed, I can change what I'm putting into it. And so then when you guys pick up these vesicles that I'm putting out, you get hold of them and you see the stuff that I put in them, and you can effectively say, oh crap, someone's stressed, someone is stressed, some other cell around me is stressed, I better freak out, I better get stressed too. And so we're interested in the mechanisms by which that happens, and you know what the signaling pathways are and all that kind of jazz. We think that actually it seemed like a really mean thing for me to do, to send you those signals for you to freak out and get stressed as well. But actually, what we and others are finding as well is that makes you harder to kill. So it's acting as a warning. I'm freaking you out, I'm sending you the signal that's making you get stressed. But actually, when you get a little bit stressed, you then get harder to kill. So if something bad is potentially coming your way, it's going to be more likely that as a result of me giving you that signal, you're going to survive it. So we think it's a survival mechanism within a multicellular organism. So we've got more and more interest in EVs over the years, and we've been doing more on them, including how they're involved in stress response, as we just discussed, but also drug resistance, which I sort of take as a type of stress response in a way. So in cancer, tumors become resistant to the drugs that you give them. So that almost always happens, that the tumor often will respond initially to the drugs, but then it becomes resistant. And so we're interested in how EVs are involved in that, but also how they're involved in metastasis and other cancer-related processes. Well, we're also interested in how they get taken up into cells, like, because if I produce these EVs, you take them up, but the question is, how do you take them up? Like, how, literally, physically, how do you as cells like take these things inside of yourself and utilize the cargo? These are really unanswered questions in the field. That's the beauty of science, is that there are always more questions than answers. So, you just have to go read a bit, and you'll find that there's a gazillion questions that need answering. So, yeah. So that's the current work that we do. If that's covers it, if that's enough.
That sounds interesting. And is there, sorry, just a quick question. Obviously, that's sort of that was to do with the modular biology of cancer, so it's concentrating a lot on cancer. But I'm guessing that has applications across other diseases as well.
The EV stuff, yeah, for sure. Yeah, for sure. I mean, so EVs generally they have potential in all kind of areas. I mean, the whole field itself is exploding. So there were a handful of papers over the last 80 years or so. Well, that's not fair. I mean, I'm digressing now. So the first experiments where we think people were using EVs, they didn't realize they were using EVs. It was back in the 40s. But realistically, the field didn't really take off until the 80s, when we started to really see that there were these things, these vesicles were being released and they had functions. And to be fair, it was really a paper in '96 which showed that these vesicles which are being released, a paper by Grassa Raposo and in her lab, that they really have a function, biological function. And so since the late 90s, early 2000s, the field has exploded, like really exploded, especially in the last decade. It's massively exploded. And so it turns out that they do have a lot of different roles and a lot of different processes. And the other thing is that they have the potential to be used as a diagnostic, because the cells release these things, and the content of these vesicles reflects the parental cell, the cell from which they came. And so they get released into every biofluid, into blood, into urine, everything, right? Saliva, tears, any biofluid you look into there. And so they could be used potentially as a proxy to see what's going on in the rest of the body. And so cancer cells, for example, there'll be, if you're unfortunate, if you have a tumor that's present, it will be pumping out these EVs, these vesicles, into the blood, probably, almost certainly. And then you have the potential then to look in the blood to see the molecular evidence, the molecular biomarker that this tumor is present. But it's not just for cancer, it's for many, many diseases have the potential to be used in this way. But also we can use them to deliver cargo. So there's a lot of interest in EVs as a therapeutic vehicle, because what we're saying is that these vesicles represent a natural way that cells can communicate with one another. I am a cell, I put stuff into this vesicle, I throw it to you guys, you pick it up and you use it. You use that cargo, right? And so that's one of the biggest barriers to a lot of these really fancy therapies that are being developed, like gene therapy and stuff like that, is that we can't actually deliver these fancy therapeutics to where they need to go to. And these vesicles are a natural way that the body does it. So there's a lot of effort being put into harnessing their power as cargo delivery units in therapeutics for many, many different diseases. It's an exciting time at the moment. Yeah, not just cancer.
Cool. Okay, so now we've also seen that you've been published in a fair number of journal articles. So is there any one publication which you remember really, really well, or do you have a favorite research paper that you could tell us about?
So I mean, there's been a few. I don't know if I could pick a favorite. I mean, I've got a few highlights in terms of the ones where I think probably the first ones, like the first paper that you get, is often quite memorable. So, and that was the PhD, and obviously with a PhD there are highs, there are lows, and then the highs are really high, the lows are really low, and so it is a real roller coaster when you do a PhD for the vast majority of people. And so when you get that paper out, it's very exciting. It's really quite something. So that one. I think the other one that stands out in my mind is probably the 2015 paper that we had, where we showed that this particular microRNA, type of non-coding RNA, was involved in drug resistance in ovarian cancer. And the reason that that particular one stands out for me is that that was the first paper that I really had as a PI, as a group leader, as a lecturer, where I had come up with all of the ideas. I had applied for the funding, I had won the funding, I hired somebody, we did the experiments, we got the data, we analyzed it, and that was the first one which really it was basically my project. So there's that one stands out as well. But I think they're all nice. I think the other, what I would say is for me, one of the really lovely papers that has been out in the last few years was a paper by a very good friend of mine, Mola Fabry, and he's now based in Hawaii. He was based in Ohio, I think, at the time. Then he moved to LA. But anyway, he had this paper. So microRNAs. I'll tell you a little story. microRNAs are these small non-coding RNAs which we think generally work by repressing a gene. Right, so they bind to the mRNA, an mRNA which is coding for some protein, and they bind to that RNA, and effectively, to cut a very long story short, they block it. They either cause the RNA, the gene mRNA, to be degraded, or to just block the translation into protein. So they repress it, and that's what we've grown up. I say grown up. I mean, they've only been around for 20 years, but that's what, for the last 20 years, we've known is that's what they do. And so Mola published this paper a few years ago now, I think it was 2010, I guess, 11, where he showed that actually they can also work in a different way. And what he showed in this paper is that they can be delivered as in vesicles, and they actually act as ligands. They can act as ligands to interact with a receptor. So we thought of them as going into the cytoplasm and repressing gene expression, but it also turns out that they can act as molecules which interact with a receptor and cause a signaling reaction, which then contributes to cancer progression. So that was a really nice one. The other one that blew my mind a bit was Laura Poliseno. So she was at the time, I think, based in New York with anyway, I can't remember. I think it was New York. And so she showed that pseudogenes, so this was a really cool paper, pseudogenes, which are genes which are like copies of other genes which have lost their capacity to code for proteins. So pseudogene, it looks like a gene, but it doesn't code for a protein anymore, either because there was a mutation in that gene or because it got copied and the promoter wasn't carried, or whatever reason, so it no longer codes for protein. But actually what she showed really beautifully was that the pseudogene the RNA that's produced from the pseudogene can actually regulate the parental gene, the original gene. And it can do so by acting as a decoy, right, as a decoy for microRNAs. So the pseudogene gets transcribed, and over here is the gene which is being repressed by a microRNA. And so there's this whole regulation thing going on. And then what happens is the pseudogene gets switched on, and it has the same regulatory region in the RNA. And what happens is that the pseudogene, which doesn't code for any protein, essentially says, the RNA says, come to me. It acts as a distraction, acts as a decoy. It causes the microRNA to come over to it, which then leaves the real gene, the real mRNA, free to be translated into protein. And so it turns out that there's this whole layer of regulation going on inside of cells, which we didn't know about. And I find that whole idea quite fascinating. So probably those.
You're very good at explaining things. I'm just gonna.
Thank you.
Okay, so what single piece of advice would you give to aspiring scientists and researchers in undergrad or secondary school who are, you know, currently discerning their career path?
It depends what they want to do, I suppose. If they, yeah, I think I would say just follow your interest, whether it's science or otherwise. I think science is brilliant. I love science. I think the power of science is just incredible. And I think the thing about science is that it doesn't just, you know, stop there. It's not just about the science for science sake. It's a way of being. It's a way of thinking. It's a whole, you know, like the scientific method is in my opinion amazing. So I would say that if you're interested in science, then go for it, get into it, because it doesn't close many doors. Right? It doesn't close many doors. So if you go into science, the skills that you gain from a science degree will stand you in good stead no matter wherever you go, whatever you do. But if you're not interested in science, then don't do it. Whatever career you're thinking about doing, follow what you find exciting, because if you don't, you're going to end up doing a rubbish job that you find boring. And don't be afraid to take risks as well, I would say. I mean, I'm about to, as you may well be aware, I'm about to change careers myself, because I've, you know, got to a point in my career where I'm thinking I've now not got comfortable, but I know what I don't feel as challenged as I want to feel. I like to fill out my depth a little bit, not too much, but a little bit. And I don't feel as out of my depth anymore as I used to, because you always get this imposter syndrome, right? You think, I can't do this, and I don't know what I'm doing. Someone's going to find me out sooner or later. And that's starting to go away, which I think is probably not a good sign. So I'm looking for something else where I can think, well, I'm not good enough to do this. Someone's going to find me out. But yeah, so don't be afraid to take risks, because you only live once, and follow what your interests are.
Yeah. And thank you so much, Dave, for this lovely talk. It's really fascinating to see the potential of whether it's extracellular vesicles and also your work also on the Human Genome Project. It's been fascinating to listen to you. Just to end on an interesting note, and as well, hopefully we'll be able to remain in touch at Brookes, but we wish you all the best in the future. But as fun reoccurring questions we would like to ask as a theme in this podcast, we would like to ask every guest who would their figure or famous person really who they find inspirational, admired, who the guest would love to invite maybe over, whether the guest is dead or alive, over dinner, to have a conversation over dinner. Anyone who was really standing out in your life, who would it be?
Yeah, and what happens at the end? Do you invite them over? Or?
Well, yeah. You, I mean, I don't know who you have in mind, but maybe post-mortem or alive now. I mean,
well, if they're dead, it wouldn't be possible. But so I, well, probably.
Probably.
So
yeah.
So I saw your email about that with that question. I was thinking about it this morning. I was thinking, who would I? And then I was thinking, I want to give you an answer that's interesting and not kind of, you know, or I'd invite Einstein for dinner. So I think, and I was thinking through. I was thinking, well, like I suppose the things that I find the most interesting are the whole, you know, the DNA, the Human Genome Project. So you could think, well, maybe, you know, maybe the early pioneers, Rosalind Franklin, you know, Watson Crick. Yeah. Anyway, maybe not Watson, but anyway. And so, and then going through like the Human Genome Project, John Sulston crossed my mind. I think he'd be great. He was in charge of the UK side of the Human Genome Project. He was the head of the Sanger. He was a really, really lovely man, actually, and very much missed. So, or Francis Collins, and there's so many. And then on the other side, I quite like space stuff. I quite like physics and so all of the whole NASA moonshot stuff. So scientists associated with that. But then I thought, you know what? I'm going to be a bit kind of controversial. I'm going to be a bit kind of. I asked my wife what she thought about this, and she was like, she initially gasped and recoiled in horror. I don't know if this is going to be very popular, and I do get that he's controversial, but I am quite fascinated by Elon Musk.
Oh, right, okay.
What he does,
Yeah.
and the dog's just going to join me. Do you like Elon Musk? So he is a controversial figure, and he does say some stuff which I just think, or do stuff which
he's a bit out there. Yeah,
he's a bit out there, and sometimes it does cringe, and it makes me, but you can't fault his drive, his work ethic is outrageous, and also he is a scientist. He's a physicist, an engineer.
And computing, I mean, he started off in PayPal and coding.
Yeah, absolutely. So but he has a very much an engineering approach, I think, to problems. But what I admire about him is the fact that he's not taking on small projects. He's taking on
the world. Yeah, gosh,
he's really taking on the world and actually trying to take it to a different world. So the fact that he's saying, oh look, he has said he doesn't want to have any money left over at the end of it. He wants to put most of it into going to Mars, and you gotta and he'll probably do it. He almost certainly will do it, and you've got to admire that kind of approach, that sort of all-in approach to
that mindset. He has a very strong mindset. Yeah.
Totally, and as I said, I do appreciate that he does sometimes say things that are controversial, but I've got a lot of time for that kind of all-in sort of.
I think it's also a good marketing mechanism, trying to say if you have to get into the front papers in some way, I guess Elon Musk is a very good choice. I have to admit, it's a very interesting.
Do you know what, I think I suppose the last thing I was going to say was think what you like about him. He's some people really dislike him. I think he's quite cool. I mean, I do think he's a bit quirky and sometimes says things that are a bit inappropriate. I don't have a major problem with that, but he wouldn't be boring.
You can have a good laugh at a couple of things over the table. You can't have someone super.
A beer and a burger would be good.
Yeah, not too bad. And well, thank you ever so much for coming on and for joining us. And we hope that all the students and current or previous students or will be able to listen into this, and there we are. Is there anything you want to address? What's going to happen in the near future where you're going towards your career or future projects you'd like to advertise? Or
well, no, as I'm starting that new role in next month actually, and so I'm quite looking forward to that. It's just a new challenge, I think every now and again you just need to shake things up a little bit and have a new challenge, which I'm particularly looking forward to. So obviously I don't know how that's going to pan out. I had a nice stable academic professor's job, which is now about to disappear, and I'm going to be going back into that kind of temporary, job market way. You don't know how long your job's for, because this is a biotech company I'm going to work for. So I don't know, they've got a certain amount of runway, and after that I don't know what happens. So but I'm looking forward to it. I'm quite excited about the challenge. And that's why I say to people to take risks and to not get too comfortable.
Well, we wish you all the best of luck on this new adventure, and thank you everyone for listening in, and we hope you have a good week. And tune in next week, where we will be joined by Caitlin conducting an interview with Emma Louise Robinson, part of the careers team, and we'll be discussing what the careers team will be offering, and also further interviews from then on. In the meantime, take care and thank you for listening. Bye now.
Bye.
Cheers. Good job, guys.